Yeah, again, thank you everyone for joining in on the call today. My name is Aadit. I'm originally from Chicago, which is where I'm here right now, but I'm a bioscience enterprise student at Cambridge in the chemical engineering department. And today I'll be talking about emerging RNA viruses, in particular, epidemics, biowarfare, and therapeutics. And so hopefully I can live up to the lofty name of my talk and the great graphic that Eva generated for us today. To kind of give an outline of my goal for today's story, on a high level, I would just want to sell you on the idea that viruses are really cool. They're not a transient thing they're going away anytime soon, and that there's a lot to explore even outside of SARS and this pandemic. But in terms of today's story, I want to start out by talking a little bit about SARS in the context of other emerging viruses and just kind of bring some interesting points and data into the picture. Talk about, well, why do we care about certain viruses and what drives us to pick and choose how and what we study in the virology space. Then talk a little bit about my basic science research that motivated this talk, and talk a little bit about how we identify some fundamental viral biology. Tell you a little bit about, well, what we can do with it, what the point of it is, and then talk a little bit about therapeutics with the goal in mind and the different approaches that we can have to treating and combating viruses. And so before I dive in, also, if anyone has any questions, feel free to interrupt me at any point. I'm happy to answer any questions or give any clarification. Okay, so SARS, right? It's everywhere today, but what's really important and interesting is that it's actually one of many emerging, not just viruses, but RNA viruses specifically. And it's something that is present or originated in Asia, but there are other viruses like different forms of flu, chikungunya, dengue, Hep C, West Nile, as you can see on this map, that are originating or re-emerging from every single part of the planet across decades, across countries, across geographies. And so in many ways, right, I think what I'm trying to bring in your mind is, well, is SARS just a virus that was in the right place at the right time? And well, we might think that our experts in the virology space knew about SARS and SARS-CoV-2 and coronavirus in general prior to this pandemic. But if you look at this, before 2020, coronavirus constituted less than 0.5% of funding in the US. So, it was on the order of $100 million when the National Institute of Infectious Disease and Allergy, as part of the NIH, was getting funded on the order of $5 billion. And so if you really think about it, SARS wasn't something that was really brought to the forefront of our minds until 2020, and maybe it was part of our minds back in the early 2000s. But really what I want to drive home here is that there were no strong indicators about SARS before this pandemic happened, and any of the viruses that I mentioned on this previous slide have the potential to create a pandemic and have a pandemic-like threat to our society. And so with that in mind, I want to introduce you to the virus that I'll be talking about for the majority of today's talk, which is a little bit more wordy and a little bit less well-known, but it's called Venezuelan equine encephalitis virus, or VEEV for short, VEEV. And I just want to bring two facts into the picture. One, so VEEV, unlike the coronavirus and SARS, it normally oscillates between mosquitoes and rodents in an enzootic cycle where it's relatively stable and it doesn't really cause epidemics or animal death. But then when it hops species into equines, into horses, it has epidemic potential where it generates high amounts of titer or high amounts of viral load in the horses, and there can be transmitted via mosquito, this epizootic vector, into humans and cause pandemic. It's primarily located right now in South and Central America, throughout a majority of countries, but it has numbers that are nowhere near SARS. It has had maybe 20 outbreaks that are relatively small and less than 50,000 reported cases, with very limited fatality. And so now, if you take SARS, for example, and the experience that all of us have had over this past year, and I show you VEEV, well, you might ask, well, why do I care about this virus? And that's the point of this message here, that, well, epidemics are actually only one part of why we care about viruses. And in the case of VEEV, and why it's actually funded by the Defense Department, research for it is funded by the Defense Department of the US, is that it is an aerosolizable encephalitic virus. And so what that means is that it can quickly or easily be aerosolized or spread in the air, And it's encephalitic, which means that it affects neuronal tissue or our brain. It has really, really detrimental, debilitating impact and pathology on our central nervous system. And so in this image, on the bottom is just normal, healthy brain tissue. And then on the top, all of this brown is death induced by VEEV. It's creating essentially mush in our brains. It's killing these cells quite rapidly on the order of days. And because of this, it was actually weaponized by both the US and the USSR during the Cold War as part of their bio defense programs, because of its potential, not naturally to cause an epidemic potentially, but also because it could be artificially induced. Right? And so there is this kind of previous message or previous history of VEEV being used in this way. But on top of that, I think that there's always hope in the virus field, and there's always different ways that we can explore and use viruses. And so something to kind of bring even more excitement to VEEV and the idea of encephalitic or viruses that infect neuronal tissue. Well, actually, just a few years ago, around the Zika epidemic, if you remember that, maybe back in 2016, researchers found that Zika virus actually has oncolytic activity against glioblastoma stem cells. And what that means is that Zika virus selectively can infect and kill cancerous glial cells in the brain. And so in theory, it could have actually a therapeutic role on an incredibly fatal central nervous system cancer that has an average median survival of about 15 months. And so with viruses like this, even though they were abused in the past, there's always hope for future utilization. You never know how we can use and leverage these viruses to learn more about our host biology or provide defense against disease. And so with that, it's always important to learn more about kind of the fundamental aspects of virus biology. And so now I want to talk a little bit about what our research project was focused on and what we did. And so in particular, what we were hoping to do is identify a very fundamental part of VEEV biology, which is what is the receptor? What is the entry molecule that is expressed on host cells or own cells that this virus uses to latch onto cells, enter and infect them successfully? And the approach we used is something I'm quite fond of. It's the throw everything at the wall and see what sticks approach in many ways. And the idea is, if you can see my mouse, you take a population of cells and you induce it with these CRISPR-Cas9 library. And you don't have to know anything about the mechanism of CRISPR-Cas9 besides it can selectively knock out different genes in our bodies. And in this library, there is a CRISPR-Cas9 guide designed for every single gene in our genome. So there's 20,000 or so genes, and so there's at least 20,000 unique CRISPR-Cas9 guides that target these individual genes. And so if you look on this graphic on the top right here, you can imagine that cell A doesn't have gene A, and cell B doesn't have gene B, and cell C might not have gene C, and so on and so forth. And so by taking this population of cells and introducing this library of CRISPR-Cas9 guides, you can generate this heterogeneous population of cells, this diverse population where each cell has a different gene knocked out. So each cell is unique from the next because it is lacking that gene. And so now I can take this cell population, transduced or presented with this library of guides, and infect it, and just see what happens. And the idea is that if I infect this population with my virus of interest, in this case VEEV, and I see what happens, the cells that survive, right, the cells that are here on the left on this top graphic, are ones that are lacking or missing some host factor the virus needed to kill the cell, right? Because all of these cells are missing one gene. And if a cell without some gene can survive this virus, that means that that gene is necessary for the virus to infect and kill the cell. And so with this strategy, we can essentially present this virus a unique population of cells that spans the entire genome and effectively let the virus help us choose which cells are resistant to the infection and figure out, well, why are they resistant in the first place? And so on the bottom is just a snippet from our specific paper where we took this library of CRISPR guides, and in this case, we were dealing with mouse cells, so it was from mice. And we introduced it to these specific cells called N2A cells. And all that's really relevant is that they're neuronal mouse cells. The reason we chose neuronal cells is, again, because VEEV is an encephalitic virus that infects neurons. And so we wanted to choose a cell type that was suited to the pathology or the cell preference of VEEV. Then we infected this population of N2A cells with these CRISPR guides with our virus, and then backtracked with sequencing to figure out, well, of the cells that survived, what gene are they missing that helped protect them? And from that, we identified this molecule called little rad3 or LDL rad3. And on the very left, what you see is this graph of the top candidates ranked by their p-value. And what this graph is saying is that before infection, you can imagine that all of the genes had probably the same number of cells knocked out, right? So going back to that previous schematic, maybe there were 10 CRISPR guides knocking out or targeting gene A, 10 CRISPR guides knocking out gene B. And so there was almost a uniform distribution of knocked out cells for each gene before I infected these cell population. But then once I infected these cells with this library with the virus, then the only cells that remain are going to be cells that are missing a gene that the virus needs to infect the cell. Some gene that when is missing has a protective effect on infection. And so what this diagram is saying is that little rad3 was most upregulated or most present in the post-infected population. So of all the cells that survived, little rad3 was missing in most of those cells. And so what that means to us in many ways is that this is now the receptor for little rad3 or for Venezuelan equine encephalitis virus or VEEV, because what this is saying is that this molecule is so fundamental to the VEEV life cycle and interaction with human cells that without it, it simply cannot infect cells, and cells can continue to survive freely without issue. And so not only do we have that experimental data from our own work, what's really exciting is that evolution almost corroborates that story. So as I mentioned in a few slides ago, VEEV infects rodents, horses, humans. And so you can imagine that, well, whatever receptor this virus is using to enter cells, well, it has to be shared across all of these animals, or it wouldn't be able to infect them, it wouldn't be able to spread on them. And so it's really exciting to see that evolutionary data corroborates this, and that little rad3 is effectively conserved at the protein level across all of these jawed vertebrates. And even outside of that, when you go to birds, when you go to amphibians, the difference is quite small in the sequence. And so little rad3 is a highly, highly conserved gene that clearly is shared across all the animals that VEEV can infect, which just further supports our hypothesis that little rad3 is the receptor for VEEV. And then as a quick detour, right? Well, you might be thinking, well, I've never heard of little rad3. This sounds really irrelevant. Well, what's the point? What's really interesting is that oftentimes these viral receptors that we identify for viruses are kind of obscure, unknown host factors without kind of this canonical role in the context of host biology. And so this is actually just a plot of papers on PubMed that mention ACE2, which is the receptor for SARS-CoV-2 and SARS-CoV-1, and little rad3, which is the receptor for VEEV. And so as you can see, before this first epidemic in the early 2000s of SARS, there was no mention of ACE2 effectively in the literature. And then even then, there was a small rise before, of course, a skyrocket in the past year, where we're seeing many, many publications on it because of the current pandemic. And so it's actually, quite common for a lot of these receptors to have really unknown function in host biology, and oftentimes really only get figured out after we identify or connect this host molecule to some kind of virus or pathogen. Okay. So now going back to studying little rad3 and proving to you that it has this role in infection. So I don't want to spend too much time on this, but just to quickly walk you through some cell culture and mouse data that we did to kind of support the idea that little rad3 is important in infection. We infected cells with 3 different types of VEEV, 1AB, 1C, and 1D, which are these 3 graphs. And these white circles are just control normal N2A mouse neuronal cells. The red circles are the cells that are missing little rad3, have little rad3 knocked out, they're not present. And then the blue circles are taking those knocked out cells that are missing little rad3 and then adding it back to show specifically that adding it back has some kind of effect. And so what's really exciting about all three of these graphs is that in these three different strains of VEEV, we are seeing this shared pattern where when we remove little rad3 from these cells, these host cells, well, now the viral titers, so the viral titers, the amount of virus over time is graphed on the y-axis, is on a log scale, so logarithmically on 10, 100, 1,000, 10,000 times lower than when little rad3 is present on the surface of the cell. And so this is just showing us that when we remove little rad3, we are seeing significant log scale decrease in virus titer, in virus amount. And the same is what's really exciting is that, oftentimes with biology, with science, it's often a difficult leap between the in vitro, the cell work, and the in vivo, the life, the mouse human work. It's really exciting to see that this same phenotype of little rad3 having this role in infection is seen in mice as well. And so here, what we did was we actually created this antibody that had or expressed little rad3, the cell receptor, freely in solution. And what we did was we injected this therapeutic, this little rad3 constructed antibody, this synthetic antibody, into mice right before infection, and saw that over time, the mice that had this therapeutic we designed based on the cellular receptor actually survived. A majority of them, 80%, 90% survived, whereas all of the mice without the therapeutic that we designed died within 10 days time after being given VEEV. And so in particular, what I mean by this therapeutic is that we created this prophylactic or post or close to exposure therapy, where on A is kind of the wild type infection pattern that you see in a mouse or a human when they get infected with VEEV, which is that this Venezuelan equine encephalitis virus is floating around in our blood, in our bodies, and it comes across this little rad3 receptor expressed on the cell surface. And it can bind to this cell receptor, and then through that enter the cell and cause severe disease and death. With our therapeutic, which is this antiviral decoy, essentially what we did was we created a free-floating version of little rad3. So now there is intrinsic competition in the host between the cell surface expressed version of little rad3 that is naturally expressed and this introduced prophylactic therapy that essentially is binding little VEEV out of solution and preventing it from entering cells by binding it up before it can get there. And so this prophylactic therapy was kind of the endpoint of our work in that it not only allowed us to demonstrate that little rad3 was the receptor and had this role both in cell culture and mice, but that we were able to design a therapeutic, this antiviral decoy, based on antibody structure to essentially propose a solution to prevent future VEEV disease in the off chance that it becomes used as a weapon or it turns into true epidemic threat. And so now moving on to goal-oriented therapeutics. So you might be asking, well, in the context of SARS, in the context of flu, I hear a lot about, well, vaccines. And well, Aadit, today you told me about your endpoint being this antibody-like therapeutic that was prophylactic, where essentially around either before or slightly after the time of viral exposure, you can introduce this therapeutic and then this decoy molecule and then bind it out of solution to prevent infection. And in the context of SARS, well, we're not really seeing the SARS therapies created by Regeneron and Vir and other companies that are meant to serve as kind of these prophylactic therapies being talked about. We're seeing Pfizer and Moderna and AstraZeneca and J&J, Johnson & Johnson being talked about for their vaccine. And that's because, well, we have to meet viruses where they are, right? And so in the context of SARS, well, by the time that these therapies came to market, this SARS was already, had turned into a pandemic. And so now it was all about preventing future infection, and it was no longer feasible to essentially say that we could prevent all present infection. But in the context of VEEV, especially early on, if we can catch it early on in the context of an epidemic, but especially in the context of biodefense, if, for example, soldiers are exposed to this or a city is exposed to this? Well, prophylactic therapy certainly makes way more sense because, well, the patients, the soldiers, the people don't have time to build immunity like in a vaccine. They need protection immediately, like we showed in this antiviral decoy. And so in that context, it would make way more sense to present a prophylactic therapy. And so the same was true in those niche edge cases for SARS patients that came into the hospital, had a lot of pre-existing conditions, were elderly or had other kind of conditions that had caused SARS to create or cause really terrible disease. And they were given this kind of prophylactic therapy to just prevent and death, prevent severe disease because they didn't have time to build their internal immunity through a vaccine. And so it's just really important to know that, even though there are some of these generalizable lessons, it's really important to meet viruses where they're at and think about, well, what context we would be working with and handling these viruses. And so with that, I'll leave you with some parting thoughts. And so if any of you here are a fan of some old school rap, hip hop, well, there's this kind of eerie line from this 2004 song that mentions SARS. And the point of that is, it's kind of weird to think about SARS being something that was only something that was talked about or had potential epidemic threat, maybe 17 years ago. And now again, it has grown to this large proportion. And if you look at this timeline, this is from year 1 AD to 2020, there's a concentration of these epidemics, these pandemics, here in the last century, and especially in the last 50 years, the last 40 years. And the point of this is that globalization increases as we continue to spread as a society. We're going to continue seeing these pandemics on top of just the existing always threat of biodefense and weaponization of these viruses. And so whether it's SARS or something else, virology is here to stay, fortunately or unfortunately, and we're always going to need techniques like this to further study and learn about these viruses and what they can do for us and how we can defend against them. And so with that, I'm going to pause there and thank you for listening in on my talk. And I'll take any and all questions that you might have. So thank you.